Welcome to Integrins and RGB motif, which is the missing subject which we haven't covered yet. So I have to switch to another window so you can see this and me at the same time, and I can switch. It's from our course, Biological Reactions of Materials, and integrins are one of the potential topics that neither of you chose. What are integrins then? They are a type of protein, so I'm kind of reading, but not fully. They're a type of protein that live in the cell wall of animals and also in plants. They're plant equivalents, and they communicate with the outside world. So for the cell, they communicate with neighboring cells and with the extracellular material, which is proteins and sugars that make a kind of gel and keep us all together. Typically, there are two subunits, an alpha and a beta subunit, which is similar to the heme protein, which you'll probably be familiar with from blood. Blood has the single heme unit, which contains the iron, and another one which doesn't, and four of those make up the heme protein. Each of those are built separately by our protein production mechanism. And in the same way, integrins have separate units, and the alpha and beta units typically hang around together. They both go through the cell wall. We'll see them in a moment. And they are liquid because they're in the cell wall. It behaves like a liquid, but 2D liquid. And they can then gather together in little clumps or spread out depending on how the internal chemistry is and the external chemistry. Typically, although they talk to the outside world, they don't exchange chemicals with outside, so they're not pores, which are typical for other things. So you'll know about the sodium potassium ion pump, for example, which pumps sodium and potassium ions, and that is normally for nerves, and they allow things through, or the water pores, which allow water through. Some of you maybe will mention a method of attacking cells, which is to insert new pores which allows too many things to get through. Integrins are not part of that system, because integrins don't let anything through. But they do allow a signal through, and to stay in the cell wall, they obviously have a hydrophobic region. This is a diagram of them. The green one is the alpha subunit, and the blue one is the beta subunit. The difference is this sulfur-sulfur cysteine bridge between two parts of the alpha subunit. The outside bit is what's going to attach to its target. It says matrix binding. On the inside, we can see it's going to bond to actin, which is the skeleton of the cell. Actin is one of the proteins in muscles. Actin and myosin are the two proteins that ratchet over each other to make you move, us move. And I know it looks a bit weird, so I have to look down to see what's going on. We can see these yellow divalent cations, which are typically calcium or magnesium, depending on which target the integrin has. It depends. Obviously, there's a big family of them, and they have different targets. The little coil section is the transmembrane section, and that's obviously the hydrophobic bit. So if we pull on it, we will move the cell wall with it. But of course, we need to bond to actin so that we can get something strong. We'll come back to that in a moment. So here is a more complicated view, because the binding can be influenced by other things. So if there's another signal, it can switch on integrin stickiness and cause it to then bind to something outside, and it can be switched off. This will also be important later. That's usually accomplished by this protein called talin, which comes along from the back and sticks on, and that causes a conformation change of the head, which makes it allowed to then stick. You can see in this picture that the two binding regions are next to each other, and they obviously can't bind to anything like that. When they open out, they can then bind. Here's a diagram from a different source. The source is down at the bottom here. The diagram is obviously a slightly different style, which is why I've left it till now, because it's a little bit more complicated. Talin comes from inside the cell, binds to the bottom, and causes a shape change in the protein, and that allows it to bond to, in this case, the extracellular matrix, the ECM. The extracellular matrix will be fibrous proteins between the cells. If we have some kind of signal from the outside, we can also cause it to switch on, and that will generate a new signal inside the cell to do something, quite possibly. Importantly, as I mentioned before, but I was getting ahead of myself, integrins are bound to the actin skeleton of the cell. If we imagine, so this is a sort of British figure of speech, that it's like nailing jelly to the wall. If you take some jelly, which is Wackelpudding in German, if we take some jelly and we try to attach it to something, it's very difficult. We can hold it in the bowl. We can't stick it to anything. We can't nail it to anything. That's because it's not got any very good places to hold it. Cells are very similar. They're a bag of liquid, and the cell wall is a double layer of soap molecules. There is nothing on there that's really strong enough to hold it up, and it should really form a liquid and just fall to the ground, really. There's no stiffness in there at all. The stiffness comes from the skeleton of the cell, which is made of actin. In order for the integrins not to just get ripped out of the cell wall, they need to be bound to the actin skeleton. That is how the cell can then move around or attach using them. So it can either pull on those integrin binding sites and move itself along the surface, or it can hold itself still against some kind of movement. So when they bind, there's a part in the cell, in the integrin, that is up here, this yellow bit at the adhesion site, that is not the attachment bit and not the actin part. It is there to generate signaling molecules. I missed a space there, so let's add one. It depends what the signal is. It could be, for some cells, due to mechanical stress, it could be activated, and it could also just be as soon as it binds, it generates a signal, or as we saw earlier, when another thing happens in conjunction, when another signaling molecule hits another measurement site, they can interact with each other and generate the signal. That allows the cell to decide, in a purely chemical way, decide what to do. So it's effectively similar to a program. If a cell sees a certain amount of stress, then it either dies, or it turns into a different type of cell, or it moves away. That can happen by pulling on the actin threads. Differentiation happens if it's a stem cell by converting into a different type of cell, and that is an active cue from the mechanical stress from the surroundings, what it's attached to. That's obviously normal, because if we have a stem cell in one of our organs, it needs to know which organ am I in, what kind of cells would it be useful to turn into. If it's a general pluripotent, so a type of stem cell that can turn into anything, it needs to know what to turn into. And one of the cues, but not all of them, is mechanical entirely from this binding. The other ones are chemical, typically. If we want to generate something like a new fetus, so we need to generate a new body, one of the problems is that they first of all form completely solid, and then they're hollowed out by the cells being removed again, and they have to die somehow, and that is one of the other functions of the binding, to tell the cells what they need to do. Getting on to RGD, so RGD is a tripeptide of arginine, glycine, aspartate. It's a tripeptide, so it's just a little bit of protein, and it's one of the major binding targets. It's found in the extracellular matrix, laminin and fibronectin, although they also have other binding sites. this is a good target for it to have because it's a short peptide, so it can attach to it, and it can be fairly common. For our topic, implant materials, and what happens if we do something, how is the body going to respond if we put in a foreign material, we can think of it as a possibility. Because if we're going to have cells adhering on our surface, they need to know what to do. They need to have the correct surface cues, and that includes the targets of integrins, includes this RGD peptide. So we can typically make some short sections of proteins, or make a protein that contains the right sections and use it as an implant material. Or we could deliberately avoid it to try to avoid cells sticking. Although it's a lot harder to do that than we would expect, because normal protein that's floating around in the cell will attach to our surface, and that will do something then that we can't control quite as easily. Binding isn't quite as good, isn't typically as good with a short section as it is with a bigger section, but it is better than if you do nothing at all. If we want to make an RGD containing surface, we can either get something to make it for us, some types of silk already contain it. One of the silk moths, but not the normal one, makes a protein, makes silk protein, which contains suitable binding sites already, so we don't have to do anything, although we could artificially add them. Spider silk can also contain it. They vary quite a lot, and we can again add it if we're going to make it using bacteria. Artificial peptides can be grown, and then we can chemically bond them onto the surface, either via collagen, which is easy to get to attach to surfaces, or directly to the material itself, to titanium, maybe to calcium phosphate, or to polymers, the idea being to get the cells to attach very quickly, because the quicker that you get integration into the body, the less time there is for something bad to happen, which is your topics and my other one. We can also, the natural proteins obviously I've already mentioned, if we put in our implant, proteins will attach to it, they will get denatured, get out of shape, and they may contain RGD motifs, and they will then tend to attract cells. This is obviously important if we want our cells to adhere, and it's obviously a question of how well is that attached then to the surface. Is it going to come off? Integrins also have some other side issues because they're involved in a lot of things. So if we take my other topic, macrophages come to the site of a wound, in this case an implant, but any wound. They are first of all activated by the activation system. So that in that case it's cytokines, chemicals go to the white blood cells and turn on the integrins. The talin turns them on from the inside. That makes them start to become more sticky, and they then stick to the walls of blood vessels, and that causes them to roll instead of flow through the center. When they're rolling, they move slower, which is good, and when they're rolling, they're also going along the side of the wall, so if there's any gap in it, they will pop out. If we have inflammation condition, one of the things that happens is the cells of the cell wall pull apart slightly to let these white blood cells leave the capillaries. So that's part of the inflammation process, part of the way that the white blood cells get cooled, manage to get out into the tissue. Obviously, it's a little bit difficult for us to deliberately mediate that, but it is an issue that's really linked, because we're always going to get some kind of inflammation. There is also a problem which we may end up accidentally causing, which is if integrins signaling doesn't happen properly, if the cells can't find anything to attach to, then they automatically send out signals to up and regulate the production of extracellular proteins, which can generate a hard fibrous matrix that we might not want. It's also a problem in a disease or known illness. So I got these pictures from this book, Molecular Biology of the Cell, which you can get online. with permission from the publisher. And from the British Journal of Cancer, obviously adhesion is particularly important in cancer because the cancer cells also have to integrate themselves into the body, and they are better or worse integrated than normal cells. So I will leave it there to see what happens. I did this all in one take to see whether that works. For a change, I usually cut these. We will talk about this sometime soon.